Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes only on YouTube. Well, we are back on another day on this channel where we're gonna be studying about the 11th class. What topic we are studying about? We are basically studying about the chapter Anatomy in the Flowering Plants. This is part 2 and this is the final part of this chapter where we're gonna be studying to the next. In the last video, if I want to recap you, then we were studying about the tissues. Now, group of tissues coming together and they are forming what system? Okay, we call it what? Tissue system. In humans, what do we call it? Organ or organ systems. Here we are calling the tissue system. Okay, so let's gonna be starting with this topic, the tissue system. Basically, first we're gonna be seeing the tissue system, and then we are diving deep into the what different part of the body of the plants look like in real terms after making a cut into it. Okay, so let's just start it. So the topic that we're going to be studying is that the tissue system. Now in this topic there are total 3 tissue systems present in the plant bodies. What are the 3 tissue systems? We have number 1, epidermal tissue system. Then we have ground tissue system, and we have vascular bundles or vascular tissue system. Now let's just talk about that, the epidermal, like we divided these 3 systems. So these 3 systems are divided on the basis of their structure and functionality. Okay, so location. Okay, so it is based on, this division is based on structure and location. So epidermal, ground and vascular tissue is on the basis of this. Moving on to the next thing, the epidermal tissues. They are present on the outer layer as an outer. So my outer layer is skin. So the skin is what? My epidermis. I will call it like this. Okay, dermis layer we used to call it. Multiple skin layers are present in the body. Same goes into them also. Okay, we're going to be studying about them in detail, but here just the crux. Okay, then ground tissue is the major part of it. Okay, where all the functions, and vascular is where the water conductions and all those things takes place. So epidermal tissue is what? The outer layer do what? It protects the internal delicate things, right? So this is what this epidermal tissue, it helps in what? It is for the protection. Okay, next, your ground tissue is the fundamental tissues, means this contains all the basic body arrangement in the plant. Okay, so this is what? This is fundamental tissue system. Fundamental body structure is being provided by this, and vascular tissue system is for the conduction of food and water. Okay, moving on, then one by one we're going to be dealing with it, starting it with the epidermal tissue. Epidermal tissue system. Now first point is that it is the outermost layer, or forms the outermost structure of the plant body. Okay, so if this is the plant, the outer white color thing you are seeing, this is what it is, actually the epidermis. Okay, when you will open it, other things will come up. Okay, right, everything has this. Next part is, it's usually single layered. Okay, usually means it could be multicellular also, possibilities is there. So when this word usually is coming, means multicellular thing can also be there. Okay, so it is what? It acts like what? Covering, outermost covering of the body of the plant. Then different part has a different types of qualities present into it. So if we see the part of the plant, so it has got different types of little modificating cells. Okay, so what it is, normally its cells are what? They're elongated compact cells are present, okay, which are arranged in the form of continuous layer. So if you talk about the cells, they are what? Elongated, compactly packed cellular spaces are almost not present. Compactly packed cell layer is there, which is continuous. This layer would be what? Continuously present. Okay, means for example, if this is the layer, it will be present like this, outermost layer of the plant body. Okay, so they are present like this. Now this epidermis is what, at certain place it is different in arrangement, and if you see that these cells are what, they are parenchymatous cell only, parenchymatous cell, and they have very less cytoplasm and more part is filled with the vacuole. Okay, with less cytoplasm present closer to the cell wall, because in center there is vacuole, and large centrally present vacuole. So if you ever had to imagine it, how you will imagine it? This is a cell. Okay, inside this cell, what happened? This is the vacuole. So vacuole is present like this. Then what other things would be present? Nucleus would be there. So I say that nucleus would be somewhere here. Then there will be what? There will be presence of the cytoplasm. So cytoplasm is present close to the cell wall, right? So this is the cytoplasm. Okay, so less cytoplasm is present comparing with the other organelles which is present into it. Nucleus and vacuole is what? Cell organelle of the plant. For now, just remember, 9th class we have studied something like that. When we're going to be studying about the cell, we will understand much better. Okay, moving on to the next thing. Then at certain places there are epidermal tissues which has certain more structures present into it, or the cells of epidermis modify little. Okay, so what I said, that certain more structures are present in the cells which are either modifications or help in certain way. Okay, so we will do what? We will see that. So in roots what happened basically? In the roots, there will be what? There will be small unicellular unbranched hair-like thread-like structure present. What is the function of that? Simple function is there, that it help in the increasing the area for the water absorption. We have talked about this cell like maturation. That is the cells from the outermost epidermal layer, they will grow. We have studied about this thing. So what happened in the roots? In the roots, root hair, these are what? Cell elongation thread like structure arises from cell. What is the reason for it? For water absorption. Okay, so increase the water absorption from the root area. Water absorption, it is present. Now these thread like structures, these roots here, they are what? They are unbranched. Moreover, the root epidermis do not have any cuticle present, means they do not have any smooth waxy layer present. Okay, which will be present in the stem or in the leaves, definitely present. Okay, so you might have seen that certain leaves are very shiny. If you have seen a citrus plant, citrus means any lime plant or these kind of fruits which are citrus fruits category, you will see that their leaves are highly shiny. Even the other plants leaves are, one layer is very shiny. That is why it is present? That is a layer called a cuticle wax. It is a wax structure. Wax is what? Oils. Okay, it's a waxy layer covering it. What is the role of it? Okay, the role of it is that it helps in the transpirational loss, because if the water layer, like if wax layer is there, what will happen? There won't be any a movement of water from one side to other side. How I will remember it? So if you take a water, okay, and on that water bottle, first you put a little bit of oil. The oil will make a layer through it. So next water, if you will add it, that water won't gonna join it. It will be what? That layer, thick layer of oil will making it what? A demarcation. Means that anything can't move through it. So we will remember it like that way. Okay, so this thing is there. So we're gonna remember that the root epidermal cells, they what? Cuticle, that is waxy outer layer, is absent. Why it is absent? Because if it would be present, then what will happen? Will they be able to absorb water? No, they won't be able to absorb water. That's the reason that they are what? Absent over here. Understood with this thing? This is all about the roots. Coming up to the next, in stems. Now stems also had like thread-like structures. Okay, also have thread or hair-like structure. Hair-like structure, but here we don't call it stem hairs. Okay, they are called as what? They are called as trichomes. Okay, called as trichomes. Now what they will do? They help in preventing the water loss. Why? Because they are what? They are covered by the waxy layer. Okay, so they do what? They prevent water loss. Water loss will occur when there will be transpiration. What is transpiration? In 10th class you might have studied that loss of water during the respiration process. Means you know na, there is stomata present on the leaf. They will open up, so carbon dioxide, oxygen comes, and the water also go along with it. Okay, so evaporate. So this thing happens. You know that you have made the water cycle also like this. You know what is transpirational loss. So trichomes will do what? That it prevents the loss of water because cuticle layer is present. Here what happened? Cuticle layer is present. Now these trichomes are multicellular. Earlier what we did, this root here, if this is the one cell, it is arising from this only. Okay, so it is what? It is unicellular, this root here. But over here what happened? If this is the stem cell, it might be like this. So this is what? Multicellular. This is also this is trichome only, but this is what? Made up of multiple cell. Understood? Now along with that, they could be branching also like this. So this is what? It could be branched or unbranched. So this thing is there. Understood? This difference between the trichome and root here is super important for you. So I'm marking it also up, because they can easily give the pointers and they will ask you that how many resembles with the root here, how many with the trichomes respectively, and you have to give option like 3, 2, like comma 2, like that, and you have to find it out. Clear? Even 1, 2, 3, 4 also they can give the things and you have to put it into the brackets. So these kind of questions will come, which will increase the time and the efficiency. So if these kind of notes are with you, it will be much easier for you. Understood? This is about the stems. Let's go towards the leaf part. The leaf has also certain modifying cells of the epidermis present. So next is in leaves. So in leaves, what will happen? The leaves have a specialized cells called as what? Stomata. Okay, it is have what? It is made up of stomatal apparatus. Okay, so these stomatal apparatus are what? They are made up of certain cells which are coming together. So imagine that in a leaf area. Okay, so if this is the leaf, this is the leaf. On the leaf, what will happen, beta? Multiple cells would be present. And leaf need what? Leaf need all the time oxygen and all those things. And you know that whenever you need oxygen, remove the carbon dioxide or anything they are using, we need what? Pores in their body. In our body, like nose is there, mouth is there. This help in what? It help in getting the air in and out, waste, right? They also need certain pore. Did we talk about any pores? No, we don't talk about any pore yet. We didn't talk about any pore yet. So here we're going to be studying about those pores and holes. What is that? Stomatal apparatus. That is a whole structure which containing the pore and the associated cells with it. So what it means that for the air exchange, there should be certain openings. Those openings are called as stomatal pores. Okay, so imagine that I am making one such structure over here. Okay, just see that. So this area I am just making up. Okay, so in this area, if I have to make it, I'm gonna making this part. There will be what? A center pore like structure present. Okay, so that pore like structure I made it. So this is what? That pore. So what I have made? I have made a stomatal pore. Okay, now this stomatal pore as what? This lining present which I have made, they are very thick. Okay, so thick linings are present. So I'm going to be making it very thick like this. They're not always open. We will understand that thing also. But these thick linings are present. Okay. Now around it, there will be cell present. Okay. Those cells are called as guard cells, because whenever there is an opening, means there is a gate, there should be one guard sitting to it. You know that every institutes where you have to go, there will be a guard present, okay, which will let only those students come and out which is the part of it or which is the needed personality, right? So those cells, we're gonna be calling it what? We're gonna be calling it guard cells. What we're gonna be calling it? We're gonna be calling it guard cells. So this is one and this is one. So two cells making what? One guard cells. So these are what? These are your guard cells. Guard cells. Now what is the feature of guard cells? Okay, can you see that this layer I have made it very thick, right? Thick layer. But this layer is very thin. So means this layer means this is also the part of guard cell. This is also the part of guard cell only, beta, because pore is formed by them. Okay, so what is happening? Imagine that this is one cell, this is one cell. If it is like this, imagine that this is one guard cell, one guard cell. So pore is formed by this like this. Pore is nothing. Okay, it is means formed by this. It's just we are making a story to understand it. So this guard cells outer layer away from the stomatal pore, it is what? It is thin layer. Whereas inside one, which is present over here, can you see this part? This is what? Thick in appearance. So this is what? It is thick layer. Okay, now these two layers are connected by certain filaments or the proteins. Okay, so I'm gonna be making that. So they are present like this. Okay, along with this, on this, there is a presence of what? There is a presence of stomatas. Because this is what? Leaf, na? Leaf cells will have stomata. So this is the guard cells are what? This is the outermost epidermal cells which are green in color. Green will be coming because of chloroplast. Okay, so they have what? They have chloroplast present. So appear green in colors. Now around these guard cells, the epidermal cells, they change themselves into what? Subsidiary cells. Okay, so around it, whatever the cells would be, they will be what? They will be subsidiary cells. So we're going to be making them. Now the close by cells which are present, they are specialized in their shape and size, and they are called as subsidiary cells. and the rest of the cells that would be present, we will be calling it what? Epidermal cells, normal epidermal cells. So here there would be other cells also. Okay, so these are what? These are the other epidermal cell, whereas these are what? These are your subsidiary cells. So we learned few pointers about how this will look like. Now multiple of them are present, and during the course of working, when they are closed, they will be like this. When they will open like this. So here I have made what? It is a open stomatal pore. This is what I have shown. Okay, now these stomatas in monocot and dicot plants, as you know that angiosperms are two types. One is monocotyledonous and other is dicot. So these two will have a different types of them. So monocot, examples like grasses, examples like gram. Okay, so here in the monocots, the guard cells are dumbbell shaped. Dumbbell shaped, whereas here the guard cells are kidney bean shaped. So this is also the fact. So how this dumbbell shape will look like this. Now around it there will be what? Your subsidiary cells. Okay, so these are subsidiary cells, guard cells and somatal pore. Here it is like this, which we have already made. And in both the cases we are making what? We are making a open type of stomatal pore or stomata. What are these? Chloroplasts. Now the next thing is, if someone will ask what is stomatal apparatus. Stomatal apparatus means it is the complex or the things which comprises of this thing. So it consists of stomatal pore plus guard cells plus subsidiary cells. This all form what? The stomatal apparatus, complete stomatal part. This you will see where? In the leaves only. Okay, moving on to the next thing, that is about we have studied about all things regarding to the epidermal tissue. The second tissue system is what? It is ground tissue system. So second is ground tissue system. In ground tissue system, what happened? That this is all the tissue removing the epidermal tissues and the vascular bundles present. So it is what? All the plant tissue minus epidermal tissue system minus vascular bundles or vascular tissue system, vascular bundles. So these things would be there. So it can be written as all plant tissue except epidermal and vascular tissue. Now if you see that what basically happens, lot of things are present inside it. How we will see it? So there is what, this is your plant. Okay, now in this plant what happened, that if I have to see that what type of cells present inside it, so what I'm going to be doing, I'm going to be cutting it off. So for example, I cut it over here and then I am seeing that. So once you cut it with the X, you will see what, like for example, if this is the plant body, if you cut this from X, what you will see, the downward portion would be like this, a circular section. Any part we're going to be seeing like that. So here we're going to see this part. So this outline, this is what, there would be a cells present over here. Okay, so this thin layer of the cells, this is what, epidermis. So this is what, epidermal tissue system. Inside it there will be what, there will be more cells present. So here there is a cell layer present like this. I'm not making the whole, I'm just showing that here it would be. Then after that more layers would be there. More different layers would be present there. Then there will be what, these are your vascular bundles. Meanwhile here more cell layers will be there. Okay, like this the whole system would be. Okay, this is throughout it will be present over here. Okay, now if you see that what we have to remove, which will give us what, which will give us the ground tissue. So we have to remove certain parts, this part not present and this part not present. These two things if not present will give us what, ground tissue system. So the other tissues which are present are of prime importance to us. So what comes inside it, let's see. So this we will remember it. We have what, this is cortex. The cortex has certain parts. What are the parts? Three parts of it. One, we have hypodermis, then we have journal cortex and endodermis. Okay, then we have what, we have pericycle, then we have what, this tissue called as conjunctive tissue, and in center we have cells that is called pith. So all of these things will be present into it. Okay, next, this names you have to remember. Okay, later on we will study about them in detail. So this is about the ground tissue system. Now this is what present over here, but if we see the leaf part, this is for the root and stem. What about the leaf part? In leaf part we have what, we have cells like mesophyll cells, which is representing what, ground tissue. There is a no pith, no pericycle, no connective. So mesophyll cells would be there, represented by the mesophyll cells only. In leaves, mesophyll cells represent ground tissue system, and these mesophyll cells could be palisade mesocyst cells or spongy mesocyst, which is a type difference of the cells. That too in the dicots. In monocots, it's all the spongy cells. This is about the ground tissue system. After that, the next thing is the vascular tissue system. So it is what? It is xylem phloem. So basically vascular tissue system is representing what? Arrangement of xylem and phloem. So whatever we have studied, we're gonna be arranging it over here. Okay, so if you remember that we have even studied that protoxylem, metaxylem, which is a part of primary xylem, and the secondary xylems form later on. Let's just see that what's happening over here. Okay, so I'm just going towards the different notes. So this is the vascular bundle, if you see over here. So one minute. So vascular bundles. So in the vascular bundles, we gonna be that is what it is basically talking about, how the arrangement of xylem and phloem is. So in the roots and stem, it will be different. Okay. Now what we gonna be doing it, actually we are seeing that stem and leaf has this thing, but only thing which is different is what? That is the presence of cambium. What is cambium? It is a tissue layer which is what? Which has the meristematic activity. Okay, so meristematic activity tissues that would be present, that will be what? Called as cambium tissue. Now why we have not talked about that, what constitute? It constitutes the ground tissue system only. Now the roots and the stems, cambium will be present, but in leaves it won't be present. First fact. Now just see that in the roots, monocot and dicots, how that will be different. If you see the monocots, there will not be any cambium present. So monocot stem also won't have the cambium present, and leaves we know that cambium is not present even if it's monocot or dicot. Moving on, in the dicot, cambium will be present. In dicot stem, cambium will be present, but in leaves it won't be present. Now if we see the monocot, in monocot there is a radial arrangement. Here also the radial arrangement of what? Xylem and phloem. When I'm saying radial arrangement, it means what? That if you see a structure, for example this, so it has what? This is the certain area. Okay, so what basically happened, that one thing is present here, other is present here, one is present here, other is here, means it is not present at the same level, but they are present in one after one in an alternative manner. So if we see that there is a radius which is very sure about it, means for example, if at this level all the phloem is present, okay, means if this is the level which is pointing out, this is the level where all the phloem is present, means phloem with respect to center has the particular radius, and alternatively at this point, which is means the depression point which is inward, okay, at a different radius, this is what your xylem present. It is called as what? It is called as radial kind of arrangement. Same, this radial kind of arrangement is present in the dicot and monocots both. The only difference in the dicot is that dicot has one extra tissue, that is cambium. Now this cambium will be present like this, here, 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 and this cambium is responsible for what? It is responsible for the secondary growth. So basically what happened, this will grow towards outward, this will go towards inward, and that will crush the things up. Next thing, in the stem, in monocot cambium is not present, dicot cambium is not present. Both has what? Conjoint arrangement. Conjoint arrangement means that they are not at the two different separate levels, xylem and phloem. They are present one after another. Now the difference would be either they will be closely packed. Closely packed means like this, that this is phloem, this is xylem, closely one after one present. Or there is the open conjoint. Open conjoint means that this is what? Presence of certain boundary between them. Who is making this boundary? This tissue, which we have to remember where it is present. Now this is called as conjoint open, and this is called as conjoint closed. Now what is open and closed means? Closed means that it won't go for the secondary growth. Open means it will go for the secondary growth. Here also it will go for the secondary growth. This is about what? This is our things. हाँ आ गया मैं 2 मिनट में. Now leaves do not have any of the cambium even in the dicots also. So conjoint, but the conjoint arrangement will be present into it like this, which we gonna be seeing it also. So this is all the crux of the vascular bundle. If you remember about this, you can remember anything. With this, the tissue system is over. Next thing that we will be going forward would be what? Would be the arrangement of the stem, root and leaf. The same thing which I have showed you, that I cut it and seeing it. So these things I'm gonna be seeing it and observing in it. That we're gonna be studying the next. So the next part of this chapter is arrangement of dicot and monocot structures. So we're gonna be doing what? We're gonna be cutting the part of the stem or the roots or the any other part of the plant and gonna see that what are the layers present inside it of the tissues or cells. Clear what we're gonna do? Let's just first start with roots. We're gonna be seeing 3 different structures: root, stems and leaf. Of what? Of all the 2 different types of classes of the angiosperm, that is monocot and dicot. So let's dig into it. First of all, we are seeing what? Roots. Roots are what? Present inside the soil. So we did what? We take out the plant root. We then cut it. How did we cut it? Imagine that this is the root. So this is the root part. So what we did? We cut like this. After cutting like this will come like what? It will come something like this structure, circular structure. So if you are seeing over here, this part of the dicot, so if you are seeing this part, it is what? Big part, small chunk which is being cut from it. Okay, so earlier it should be like this, but they have this whole 4 part. Able to recognize it, able to imagine it. Okay, so this is it. So now if you see the dicot stem and the monocot stem, we're gonna be learning the layers from outside towards inside. Means outside is this layer, okay, and the inside is this layer. So if someone will ask like what is the outermost layer of the plant body, it is shown by epidermal tissue system, okay, and the innermost is the pith, this center part. Whatever the cell is present over here, we call it what? We call it pith. Clear? In roots and stems, not in the leaf. Leaf things are different. You will remember this, one important question it is. Now if we see the dicots and the monocots, now I'm telling you we're gonna be seeing the outside towards the center thing, as well as we're gonna be seeing the difference between monocot and dicot, and we have divided the whole thing into 3 parts. What are the 3 parts? What is the how the epidermis tissue look like in each of them and how different they are from each other. Then crown tissue, what are the layers present in each and how different they are from each other. And third is the vascular bundle. So the things which we have studied in the beginning, we're gonna be utilizing it that way. So if you will prepare this way, that would be much easier. Then comes what? If you see the dicot, let's see that outermost layer is this one, which has the what? These hairs. These hairs are representing what? These hairs are representing the root hairs. So this is unicellular hair, and this layer is called as what? Epiblema. Then inside it, this area, this green color areas where different types of cells are present. If you see, they are representing what? Cortex. Now this cortex has different layers present. So there will be what? Just below the epidermis, we call it hypodermis. Although it is not much demarcated over here, but if you see that the cells in this layer are much more compactly packed compared to here. So here the intercellular space is present in the journal cortex area. Then the innermost layer is this of the cortex, that is endodermis. Now coming inside of the endodermis, there is what? Pericycle, this layer. Inside pericycle, you found this area. Okay, this area looks little different because if you see that there is what? The root is there. Root means their vascular bundle will be what? We have just discussed, that is radial. So this yellow color thing you can see, yellow cells, this is representing phloem. Whereas this thing is representing what? Xylem. And over here also, if you see, what, like protoxylem or endoxylem, if you remember, we have even studied that how the proto and metaxylem is present, that tells us that what kind of arrangement it is, exarch or endarch. You remember in case of roots, we have what? Rx, that is exarch. Exarch means that interior there, sorry, in the center there will be what? Metaxylem, and outside there will be protoxylem. Okay, so if you see that here the metaxylem is present in the center, protoxylem is present outside, and since metaxylem would be what? The later forming ones, so it would be mature enough, okay, because it has more things, so it is shown as the bigger ones. Protoxylem is this one, smaller, which is present towards the periphery. This also we can understand or remember from here. Now in between these tissue, these layers are called as what? Connective tissue. And in the center we have the pith, which is smaller in appearance. Okay, when we comes to the monocot part, there is a root here, there is the epidermis, there is this cortex, there is the endodermis, there is the pericycle. But over here, if you see inside this area, here there is what? Multiple of these xylem present. Of course, phloem is present what? At this area. There also what? Radial arrangement only would be there. So phloem is somewhat here, the white color dots which I have just shown it. Whereas your xylem is shown in this way. But if you see that here there is 4 only, but here it is more than 4. So this condition is called as polyarch. Means multiple of them are present, means more than 6 of them present. And the center is what? Your pith, which is again been demarcated. Let's see that in terms of the tissues what we have studied. You know that you can see that now. If you have your NCERT book, you can open it and you can analyze it. If you see the epidermis tissue, it consists of epidermis layer called as epiblemma and the unicellular hairs. Here also it is same like dicot. So no difference. You will remember the fact. You can put the wordings with each other. Then comes the ground tissue. It consists of cortex, which is made up of several layers. Then endodermis. All this is parenchymatous, made up of parenchyma cells. Endodermis is the innermost layer of the cortex, and it has a Casparian strips. What is Casparian strip, or does every cells has it? No. Few cells do not have Casparian strips, but most of the cells has Casparian strips. It do what? It makes the flow of water constricted. Because you know that xylem is closer present, so xylem transport lateral conductions would be there, means the horizontal or conductions, radial conductions will be there. So to regulate it, and the minimum amount of water goes out, okay, towards the outside area, we call it. Now this area is also defined. If you see that this area is called as stellar region, whereas this is called as extrastellar region. So this is the stellar region, extrastellar region. Okay, moving on, the pericycle, it is few layers. It gives rise to the lateral roots and the various, like now the very important thing. Lateral roots is not same thing with the root hair. Root hair is formed by the epidermis. It's a unicellular. Lateral roots are what? These are secondary or the tertiary roots, which is a part of a growth pattern. Okay, since lateral roots are seen in their cases, since in the monocots, what do you normally see? You see the fibrous roots, no lateral root systems there. Okay, so pericycle, it is important, which give rise to the lateral roots, means the growth pattern is there, and vascular cambium formation is also taken place by the pericycle only. Then the pith, it is small and inconspicuous. Small inconspicuous means not visible, not appreciable. It is very small over here. Okay, whereas this is bigger compared to this over here. Now, if you see the connective tissue, it is what cells present between the xylem and phloem. It is seen here. Here, everything is same. All of these things are same except the pith area and the lateral roots formation. Now, the pith over here is large, well developed, and conspicuous. Means you can easily identify it. This thing you have to remember. Okay, this is the major part where the question can come. Okay, moving on to the next and the last most tissue part of the roots would be vascular bundles. So I have already told you here we have seen what 2 to 4 xylem phloem patches will be there, and there is a cambial ring will be present, because these are what? These are dicots. In dicots, cambium is present. How it will be present? Here, here. So this is how the cambium would be present. Okay, so this would be the growth like this. It will growth like this. Okay, so things will be crushed. So pith will be completely crushed because of it. So overall, that's why it is said that pith is inconspicuous, because what will happen if there is the secondary growth happening in this way? So it will take away this space. So that's why it Pith is completely crushed because of it. So if we have to write it, we can write it. So wait, where is, due to which pith is completely crushed by secondary xylem and phloem. Okay, this we will remember. Next, here the vascular bundles, they are more than 6, so they are polyarch conditions. No cambial ring present means no secondary growth is seen in their case. Easy. These are the points which next would be stem out of it. Now the stem, you know that this is a stem. If you cut it transverse section, what you will get, you will gonna see something, the circular arrangement. Outermost layer would be what? It would be called as epidermis. So here also epidermis will be there. Now here you know that what would be present, there would be presence of the stem hairs called as trichomes, which are seen over here. Now in dicot there will be trichomes present, but in monocots no such trichomes are present. First difference. Moving on to the next one, if you see the layers over here, these are the layers. Here if you see that it is what, same kind of things which we have studied in the root case. So this part is cut and shown it over here. This is what center, this is the pith, very big, conspicuous over here. Here no such pith thing, you can't even distinguish. Let's just talk about it one by one. So we have what, epidermal here present like this. Then this is what, epidermis. Below epidermis there will be what, cortex present. Cortex has 3 layers present: hypodermis, general cortex and the endodermis. If you see the hypodermis, here it is hypodermis present. Now here what is the special part of is that hypodermis of the dicot is made up of collenchyma tissues. Okay, so this is what, collenchymatous tissue. Then comes what, then there is a presence of what, then there is a presence of your general cortex. Now this your general cortex has what, it has little bit of patch of sclerenchyma, little bit, most of them are the parenchymatous. Then comes this layer, endodermis. This endodermis is what, this layer, and this is what, little wavy in appearance. Okay, so this is a characteristic thing present over here, that the endodermis is wavy in appearance. After that endodermis, we have what, inside more in patches, this pericycle. Okay, so the pericycle is present over there in patches. Okay, and this patches appears what, lunar in appearance, means this kind of, these patches are present throughout it. Then inside pericycle there will be what, vascular bundles present. So here if you can see this area is what, vascular bundles present. Now this green thing which you are seeing over here, this is representing what, this is representing the your basically what, the cambial rings. Okay, over here, and this is what, this is your xylem phloem arrangements which are present over here. And in between these layer, earlier we used to call it what, connective tissue, here we call it medullary rays. Okay, and this is what, in depth shown to you. Now if you see that here, what would be the protoxylem and metaxylem arrangements? The protoxylem would be present on what, it is present on the inside, metaxylem is present on the outwards. So this is what, stem. If you remember, stem has what, it has endarch kind of arrangement, means center will have what, your protoxylem. So look, center is over here, will the, where the pith is, there is will be the center, protoxylem present closer, this is present towards outside. Understood? Let's come towards the monocots. When we are seeing the monocots, if you see that epidermis is there but no root hairs, sorry, shoot hairs would be present. If you see that multicellular, multiple cuts are being shown over here, here nothing is like that. So epidermis do not have. As you go inside, there is a hypodermis and there is ground tissue. This whole thing is ground tissue, but there is no layer compartmentalization. And these are what, these are the scattered vascular bundles. And these vascular bundles has what, central water holding capacities present inside it, and they appears like this. So face like thing is being seen in their cases. Okay, let's just study about it. So if you see the tissue system, so the tissue system, we have to study about epidermis, ground tissue and the vascular bundle. So epidermal tissues, first you will see it has a thin layer of cuticle present, roots do not have that. You know that why. Then stem hairs and trichomes are present, there are many. Few stomata may be present in the early stem. Here epidermis is present, no trichome is present, and rest stomata may be present. thin layer of cuticle is present. Then comes the ground tissue. The cortex is divided into three parts. What are they? Hypodermis, cortical layers, general cortex, and the endodermis. If you see the hypodermis, there are few layers and column chimetus, and it provides the mechanical strength to the young leaf. We have already discussed about these facts in the first part of this video. Then this is the outermost layer. Then the cortical layer, they are parenchymatous. We have already discussed about it when we are discussing about the diagram. Endodermis is the innermost layer and it is rich in starch. That's why it is called a starch sheet. Sheet is what? A covering layer made up of what? Starch. So we name it like that. So this is the most important over here. Where is the Casparian strips present? In the roots of the endodermis cells. That is the difference where the questions came. Now the next thing is over here, pericycle is also there. This is the inner to the endodermis, above the phloem, and present is semilunar patches. I showed you like this. They are made up of sclerenchyma. So this is very important fact. This is collenchymatous, sclerenchymatous, parenchymatous. Endodermis also parenchymatous. Then the medullary rays is present between the vascular bundles, means xylem phloem. What is present? Not the xylem phloem, but the different vascular bundles. The space that is present in the root, we call it connective tissue. Here we call it medullary rays. And the pith is there, which is large and conspicuous in their case. We have already seen it. Then comes the ground tissue of them. Now we know that their ground tissue is what? Just hypodermis is there, and then the general cortex is there. No clear distinction between pericycle, endodermis, pith, medullary rays, or anything present. Their hypodermis is sclerenchymatous. We have already discussed about it. Then the large conspicuous ground tissue made up of parenchyma, but no clear large division is present, means no pith, no ground tissue, no endodermis, no pericycle, medullary rays could be identified. The last is the vascular bundles. If you see the vascular bundle in their cases, they are what? They are arranged in the ring form. So you can see that this is the ring of it. That's the red color things. So they are present in the forms of rings seen only in the dicot stem. Conjoined open with the enlarged protoxylem. This we have already discussed. Open means what? Open to the growth, because there is what? Cambium present to it, which we have already shown and marked into it. This thing here in this green things. Then comes this vascular bundle over here. They are scattered everywhere. Now if you see that outer, there would be what? This area would be smaller ones. Here will be larger ones. So in the scattered everywhere, and they're surrounded by the sclerenchymatous bundle sheath. They're conjoint and closed. We know that. Then peripheral vascular bundles are small. Central are the large. With the diagram only we can see phloem parenchyma is absent. Here there is no phloem parenchyma present in case of monocots. This is very important fact. Then water containing cavities. Now tell me that what is the role of phloem parenchyma? To contains what? To contains the food. And what is the function of parenchyma? To contains the food, right? If you know that everywhere there is what? Parenchyma present, they don't need any special types of abundant food could be easily derived from them. So that's why phloem parenchyma is absent. Okay. Then water containing cavities, I have already told that present between them. Okay. With this, the stem is over. I hope this thing is understood. If you understand the root, stem is not tough. Only the learning things would be there. Distinguish, and then you can remember the facts. Then comes the leaves and the last topic of it. So the dicot leaf, monocot leaves. Now the leaves, if you might have seen, the leaves have something like what? This kind of structure. Like for example, this tablet thing. So think like if this is the leaf, this part is the leaf. Now in the leaf, there is what? Upper part, lower part. You might have seen that the certain leaves have one part, the upper part mostly the darker green, lower part is lighter green. Why that so? Because upper part has more chloroplast, lower part has less chloroplast. You have even seen that upper part is shiny, lower part is not shiny, it's soft. Why? Because upper part has what? It has cuticle layer present onto it, lower part not present. Why the cuticle layer present? To reduce the loss of water from the transpirational loss, means this part of the leaf would be closer to the sun. That's why the more chloroplast is there, so that they can make more food compared to this one. Alright, so this thing we have to understand. Now this leaf upper and lower part, with respect to animal kingdom, what we used to say that the part which is closest towards the sun, we call it dorsal, right? Here the part away from it towards the sun, we call it ventral. So the leaf would be what? Dorsoventral in case of dicot. So we will remember D for dicot, D for dorsiventral. Dorsiventral means what? That leaf would be present like this. For example, this. But the same thing would not be in the monocot. Monocot would be isobilateral, means equal on the both sides. Their leaves are present to the plant, like if this is the plant body, it will be present like this. So both the side would be same color and there won't be much distinguishing. Okay, so if you see that, this is how they will look like. Now remember that they have what? They have epidermis, they have their ground tissue, and they have their vascular bundles. Okay, but their ground tissue do not have layering presence in the leaf, because leaf is so thin, how the layers would be present, right? So epidermis would be what? Both the sides we will call it epidermis. And here very important is that, think about it. If this leaf is like this and sunlight is completely falling on it, what will happen? If the sunlight is there, more water evaporation will be there. But if we have to reduce it, what God can do? First of all, God can do what? They can make leaves like this. So God said that, okay, I will give that feature to the monocot. What here? So what God did, God took out all the stomata from here and put it on the downside, where the less sunlight will be present, because most of the leaves will be like this. So here stomata is less present, here stomata is more present. So means all the water loss or the oxygen exchange which is needed and the part of the stomata can be done from here with the minimal water loss. Here, no. Okay, so this thing is there. Now with respect to that, we call the surface not just dorsal and ventral in case of plants leaves, we call it adaxial and abaxials. So adaxial is that surface which is dorsal or the upper surface. So ventral surface will be called as abaxial. How you will remember that? Adaxial, D is coming here, D for dorso, D for dicot. Okay, so there is what? Dorsal side of the leaf where the less number of stomata are present, more green would be what? That would be your adaxial surface. Abaxial surface would be vice versa, opposite. Okay, then we have two different types of cell layers. Okay, this is the epidermis cell. Then we have what? This cells which is long are called as long columnar cells are the palisade cells. Okay, so these are what? Palisade mesophyll cells. And these circular cells which you can see, these are what? These are spongy mesophyll cells. So two different types of cells are present in the dicot. Then this area, can you see that? This is what? This is the xylem phloem bundle present inside the bundle sheet cells. So there are what? There is a bundle sheet cell present around it. Okay, coming on to the monocots. Monocots, like both sides stomata present, so equal abundance is there because they are present like this. Okay, they won't have any such restrictions, right? Then there is what? One is the adaxial surface, one is the abaxial surface. Now if you see that there are what? These type of tissues present. These are what? This is all the spongy tissue. This dots are what? These are the chloroplast. Okay, so these are only spongy tissue present. No palisade mesophyll tissue would be present over here. Then these are what? These are the bundle sheath cells and xylem present. This is what? Your phloem present, this area. Okay, so xylem, phloem, every cavities are being made. Let's just see that in terms of distinguishing. So three tissue system present, here also three tissue system present. So epidermis layer we're going to be studying. Two surfaces are there, upper surface, lower surface, more stomata, lax stomata. Wait a second. Here we have written something wrong. This is not more, this is upside down. Okay, it will be like there will be, this would be this would be this. Okay, because to reduce the loss of water. Okay, then here both the surface has even number of stomata. Next thing is that if you see this, between the upper and lower layer, mesophyll cells are two different types. Both carries photosynthesis, palisade and spongy. Their parts I have told you. Okay, now the next is vascular tissues. So vascular tissues are what? They have reticulate venetions present into it, and vascular tissues are present over here in the bundle sheath cells. Okay, this you know that reticulate venation, what variations would be present so that you can identify it. So vascular tissues will be present in those venetions only. Okay, next is the epidermis. The monocots, they have both the surface equal. There is a presence of bulliform cells. Now you know that they are present like this, means both the sides stomata is there, water loss will be there. To reduce from the water loss, they have what? Bulliform cell. Here you have to remember that cell, CEL, they are colorless, empty and large cells. So what happened basically, that they are very turgid, they have water present and they are very much what? stiff present. If something is stiff, it would be straightly stranding. But what if the water will be evaporated? Certain period of time, water will evaporate because so much of heat will be there. What will happen? The cell will shrink. When they will shrink, they will pull the other things closer, and it will do what? Rolling or curling of the leaves, which will reduce the surface area of the leaf. And as the surface area of leaf is reduced, what will happen? Transpirational loss will also reduce, because only few stomata will left in front of sunlight. So this is what? A way to prevent the water loss. Mesophyll cells are not distinguished into placid and the spongy cells. All are spongy cells. Parallel venetions are present. With this, your topic, that anatomy of the root, stems, leaf of monocot and dicot, is completed. With this, everything which is needed for your NEET 2026 is completed. Of course, certain questions we will be practicing, and we will try to understand more very soon once the syllabus is completed. So if you like this video, give it a like, share it with your friends, share it with your classmates, share it with your old school friends, and don't forget to subscribe me, because that motivates me a lot. If you want more such kind of things, let me know in the comment sections. More reels, more shots, more videos would be coming up in the near futures. We will be trying to complete our syllabus before November, and after that we will be coming up something very special for all of you. So that's all for this video. Thank you.